mitosis and meiosis. Two words that sound almost the same, look almost the same on a diagram, and get mixed up on more biology tests than just about anything else. But they do two completely different jobs, and once you can see the difference clearly, you'll never confuse them again. Here's the big idea before any of the details. Both mitosis and meiosis are ways a cell divides, one cell becoming more cells. That is the only thing they truly have in common. What makes them different is why the cell is dividing, and what kind of cells it makes at the end. Get that, and everything else falls into place. Let me put it in one line you can hold on to. Mitosis makes body cells, the cells that build and repair you. Meiosis makes sex cells, sperm, and eggs. That single difference in purpose is what drives every other difference we're about to walk through. So keep it in your head the whole time. Mitosis for your body, meiosis for reproduction. But before we split anything, we need to talk about what's being divided. Chromosomes. A chromosome is a long strand of DNA, your genetic instructions, coiled up tightly so it can be moved around neatly. Humans have 46 of them in almost every cell, and they come in a very specific arrangement that matters for this whole story. Those 46 chromosomes are really 23 pairs. One member of each pair came from your mother, the other from your father. The two chromosomes in a pair are called homologous chromosomes. They carry the same types of genes, a gene for eye color in the same spot, for example, but not necessarily the same version. Think of them as a matching set. Same categories, different details. Now, before a cell divides, it makes a full copy of every chromosome, so there's enough DNA for 2 cells. When a chromosome is copied, the original and its copy stay attached at a point called the centromere. These 2 identical halves are called sister chromatids. This is the classic X shape you've seen in every textbook. 2 identical copies joined in the middle. Careful here, because this is the #1 thing students mix up. Sister chromatids are identical copies of the same chromosome. Homologous chromosomes are a matched pair, one from each parent, similar but not identical. Same and identical versus matching but different. Hold on to that distinction, because it is the key to this entire topic. 2 more words, and then we divide, I promise. A cell that has the full set, both members of every pair, all 46 in humans, is called diploid. A cell that has only half, one from each pair, 23 in humans, is called haploid. Diploid is the full deck of cards. Haploid is half the deck. Keep those 2 words handy. Let's start with mitosis, the simpler of the 2. Its job is growth and repair. Every single time you heal a cut, grow taller, or replace worn-out skin, mitosis is happening. It takes 1 cell and makes 2. And here is the crucial part. 2 cells that are perfect, identical copies of the original. The best way to picture mitosis is a photocopier. 1 cell goes in, 2 identical cells come out. It begins with a stage called interphase, which is really just the cell getting ready. The cell grows, does its normal daily job, and copies all of its DNA. So by the end of interphase, every chromosome is 2 sister chromatids, ready to be pulled apart. Then the division itself begins, in 4 main phases. The first is prophase. The DNA, which is normally loose and stringy, coils up tightly into the compact chromosomes you can actually see under a microscope. The membrane around the nucleus breaks apart, and tiny fibers begin stretching across the cell, getting ready to grab hold of the chromosomes. Next is metaphase, and this one is easy to remember. Metaphase, middle. All of the chromosomes line up in a single file, right across the center of the cell. The fibers attach to each chromosome from both sides. This is the cell carefully lining everything up before the big split. Single file, straight down the middle. Third is anaphase, and the trick there is anaphase, apart. The fibers pull, and the sister chromatids, those 2 identical halves of each chromosome, are dragged apart. 1 copy heads to each end of the cell. When this is finished, each side of the cell has a complete, identical set of chromosomes waiting. Last is telophase. The chromosomes arrive at the 2 ends of the cell, a fresh nuclear membrane forms around each set, and the DNA relaxes back into its loose, stringy form. Then the cell pinches in down the middle and splits into 2, a final step called cytokinesis. 2 cells and mitosis is done. And here is the result of mitosis. 2 daughter cells, each with a full set of 46 chromosomes, each a perfect genetic copy of the original and of each other. Same chromosome number, identical DNA. That's exactly what you want for growth and repair. You don't want your new skin cells to be surprises. You want reliable, faithful copies every time. Now for meiosis, and this is where it gets more interesting. Meiosis has a completely different mission. Its job is to make sex cells, sperm in males, eggs in females. And sex cells come with a special requirement, one that changes absolutely everything about how this division has to work. Think about it for a second. If a sperm cell had 46 chromosomes and an egg had 46, then when they combined to make a baby, that baby would have 92. And the next generation would have 184. The number would double forever. So sex cells cannot carry the full set. They need exactly half 23 in humans, so that when sperm meets egg, you land back at 46. Meiosis is how the cell cuts that number cleanly in half. To pull that off, meiosis does something mitosis never does. It divides twice in a row. 2 rounds of division called Meiosis I and Meiosis II. And instead of ending with 2 identical cells, meiosis ends with 4 cells, each carrying half the chromosomes and each genetically unique. Not copies this time, originals. Let's walk through it. Just like before, the cell starts by copying all of its DNA, so every chromosome is now 2 sister chromatids. Then meiosis I begins with Prophase I. But something brand new happens here that never happened in mitosis. The homologous chromosomes, the matching pair from your mom and your dad, actually find each other and pair up, lying right alongside each other. And while they are paired up like that, they do something remarkable. They swap pieces. The matching chromosomes physically trade segments of DNA with one another. This is called crossing over, and it is a huge deal. It means the chromosome you eventually pass on is no longer purely your mother's or purely your father's. It's a brand new mixture. This is one of the main reasons you are not identical to your brothers and sisters. Next comes Metaphase I. Now the chromosomes line up at the middle, but notice the difference. They line up as pairs, not in single file like mitosis. And which side of the cell each pair happens to face is completely random. This randomness has a name, independent assortment, and it is a second source of variation. Every pair essentially flips a coin for which parent's chromosome goes which way, then Anaphase I. And here is the single most important difference between meiosis and mitosis. So really lock this in. The pairs are pulled apart, but the sister chromatids stay together. It is the homologous chromosomes, the whole matching pair, that gets separated, one heading to each side. In mitosis, sister chromatids split. In meiosis I, the pairs split. That is the very heart of the difference. The cell then divides, and now we have 2 cells. And already each of these 2 cells is haploid. It carries only 23 chromosomes, 1 from each original pair, instead of the full 46. The chromosome number has officially been cut in half. But we are not finished yet because each of those chromosomes is still 2 sister chromatids joined together in the middle. So meiosis II happens, and here's the good news. This part looks almost exactly like mitosis. In each of the 2 cells, the chromosomes line up in single file again, and this time, the sister chromatids finally separate, pulled apart to opposite ends. Each cell splits once more. You can think of Meiosis II as the cleanup round that pulls those last identical copies apart. And now, the final result of meiosis. 4 cells. Each one is haploid, carrying 23 chromosomes. And thanks to crossing over an independent assortment, each of the 4 is genetically unique, a different shuffle of your mother's and father's DNA. 4 unique half-sets, each one ready to become a sperm or an egg. Okay. We have built both processes from scratch. Now let's put them head-to-head because this exact side-by-side comparison is what your exam is going to ask you for. Same kind of starting cell, 2 completely different outcomes. Let's line up the differences, one at a time, so they're crystal clear. Difference 1 is purpose. Mitosis is for the growth and repair of your body. Meiosis is for making sex cells for reproduction. Difference 2 is the number of divisions. Mitosis divides 1 time. Meiosis divides 2 times, and that extra division is exactly what allows it to cut the chromosome number in half. Difference 3 is how many cells you end up with. Mitosis makes 2. Meiosis makes 4. Difference 4 is a big one. Are the cells identical or unique? Mitosis makes identical copies, essentially clones of the parent cell. Meiosis makes genetically unique cells, where no two are ever exactly alike. Difference 5 is the chromosome number. Mitosis keeps it the same. The daughter cells are diploid with the full 46, just like the parent. Meiosis cuts it in half. The daughter cells are haploid with 23. So a full set stays a full set in mitosis, but a full set becomes half a set in meiosis. And difference 6 is the sources of variation. In mitosis, there is no crossing over and no shuffling, because the entire point is to make faithful copies. In meiosis, crossing over and independent assortment deliberately mix everything up. Mitosis protects the sameness. Meiosis manufactures difference. That contrast really is the soul of the whole comparison. Now let me save you some points because there are a few traps students fall into every single year. Trap 1 is mixing up which process makes what. So say it with me. Mitosis makes body cells. Meiosis makes sex cells. If you walk away remembering only one sentence from this video, please make it that one. Trap 2 is forgetting that meiosis has 2 divisions, not 1. That second division is the entire reason you end up with 4 cells and half the chromosomes. Trap 3 is the sneakiest of all. In Meiosis I, it is the homologous pairs that separate, not the sister chromatids. Those chromatids do not split until Meiosis II. And Trap 4 is the vocabulary trap we flagged right at the start. Sister chromatids are identical copies of one chromosome. Homologous chromosomes are the matching pair from your 2 parents. If a question asks what separates during a given phase, you absolutely have to know which of the 2 it's talking about. Slow down and picture it before you answer. Let's step back for a moment, because this is not just diagram memorizing. This is the reason you exist and the reason you are unique. Every one of us began as a single cell, a sperm and an egg, each carrying a unique, shuffled half set of chromosomes, combining into a brand new full set that had never existed anywhere before. That is meiosis handing you your one-of-a-kind combination. And from that single cell, mitosis took over, copying it faithfully, again and again, billions upon billions of times, to build every part of your body. Meanwhile, the variation that meiosis creates is the raw material that evolution works with. Without that constant reshuffling, every generation would just be a copy of the one before it, and life could never adapt or change. People pieced all of this together over more than a century. In the 1880s, a German scientist named Walter Flemming sat at a microscope watching cells divide, and he gave mitosis its name. Around the same time, August Weismann reasoned that sexual reproduction had to involve a special division that halves the chromosomes. He essentially predicted meiosis before anyone had clearly seen the whole thing. And it ties directly back to Gregor Mendel. His famous laws of inheritance, the way traits get shuffled and passed down through generations, are really just the visible result of what meiosis is doing with chromosomes down at the cellular level. Independent assortment, that coin flip we talked about, is Mendel's law playing out right inside the cell. Different discoveries, all the same beautiful machinery. All right, let's pull the whole thing together. Both mitosis and meiosis are ways for a cell to divide. Mitosis takes one cell and makes 2 identical copies, each with the full set of chromosomes, and it does this to grow and repair your body. It is a single division, and its entire purpose is to keep everything the same. Meiosis takes one cell and makes 4 unique cells, each with half the chromosomes, to be used as sperm or eggs. It divides twice, not once. It uses crossing over and independent assortment to make every one of those 4 cells different. And when 2 of them, a sperm and an egg, finally combine, the full chromosome number is restored in a brand new individual who has never existed before. For your test, lock in these 5 things and you are covered. 1. Mitosis makes body cells. Meiosis makes sex cells. 2. Mitosis makes 2 identical diploid cells. while meiosis makes 4 unique haploid cells. 3. Mitosis divides once, meiosis divides twice. 4. In Meiosis I, the homologous pairs separate, while in Meiosis II and in mitosis, the sister chromatids separate. And 5. Crossing over and independent assortment happen only in meiosis, and they are what create genetic variety. Nail those 5 points, and any mitosis versus meiosis question on your exam is yours. Here's a quick memory trick to keep the two straight. Mitosis starts with mite, so think making identical twins, 2 identical cells. Meiosis starts with me, so think of it as making the unique cells that build a one-of-a-kind you. And it makes more of them, 4 instead of 2. A little silly, but that's exactly why it sticks. So there it is, mitosis and meiosis. They look almost the same on the page, but one is a photocopier, keeping your body running with perfect, reliable copies, and the other is a shuffle and deal that creates brand new life and makes you unlike anyone who has ever lived. Same starting cell, completely different missions. 2 words that sound almost the same, but now you know exactly why they are not. If this finally made mitosis versus meiosis click for you, hit that like button so more students can find it. And subscribe, because your next topic is coming, and we'll make that one just as clear.